Welcome to biology. In this video, we are going to introduce some of the terms associated with the study of genetics, and we are going to talk about how to predict the phenotypes of offspring by using a monohybrid cross in a Punnett square. More about that in a moment. To begin with, heredity is the set of characteristics that are received from parents, so what genes are passed down from one generation to the next. And genetics is the study of heredity, or the study of these family trees, so to speak. Now, within the study of genetics, we're going to use this term called allele. Now, within the genome of an organism, you have the genes, or a section of DNA, that codes for a specific trait or a specific protein. Now, not all genes that code for, say, eye color are all the same. There are some alternate versions of these genes, and those are called alleles. So an allele would be the version of the gene for eye color that codes for, say, blue eyes, versus the allele for the gene for eye color that codes for brown eyes. So the allele is the version of gene that is passed from parent to offspring, and it's represented by one single letter. Now, if it's a capital letter, it's representing the dominant allele, and if it's lowercase, it represents the recessive allele. Now, the dominant form of the gene is the trait that is expressed when there's only one copy of that allele present. So we use a capital letter to represent the dominant form. The recessive form is a gene that's a little bit more timid. It is not expressed unless there is no dominant form of the gene available. So it is repressed or hidden by the dominant gene, and we represent that with a lowercase letter. Now, we also use these terms genotype and phenotype. We also use the terms heterozygous and homozygous. The term genotype is talking about the actual DNA inside of an organism, whereas phenotype is the physical appearance of that organism. With genotype, inside of each organism, you have two copies of DNA. You have one full set of DNA from the mom and one full set of DNA from the dad. That's why we use two letters to represent the genotype. We represent the letter you got from your mom and then the letter you got from your dad. These are either two capital, a capital and a lowercase, or two lowercase letters. Now, phenotype is again the physical characteristics, and these are usually described with words. So the color of the eyes, or purple flower versus white flower. Homozygous and heterozygous are ways to describe the genotype. We could have a homozygous genotype, meaning that both copies of the gene are for the same allele. So we would have two copies of the dominant allele, or we could have two copies of the recessive allele. We say that a homozygous individual is a purebred, and they are purebred because they are pure and only have one type of the allele in their DNA. Now, a heterozygous individual is a hybrid. Remember, hetero means different, so they have one copy of the dominant and one copy of the recessive allele for that particular trait. Now again, the way that you inherit these traits is from the eggs and the sperm joining together to make that fertilized egg. But when the parent is making the sperm or the eggs, those chromosomes are randomly separating during meiosis. So those gametes contain one version of the dominant or the recessive allele depending on what that parent had. So if the parent was heterozygous for a particular trait, it is totally random on if the dominant or the recessive copy of that gene is showing up in each egg and sperm cell. So the offspring randomly receive one allele from each parent. The other law that you need to know is the law of independent assortment. This is saying that inheriting something like eye color or plant height is totally separate from inheriting a different trait. So each trait is inherited or randomly separated out. So they're inherited separately. Now back to our genetics storyline here. Gregor Mendel is the father of genetics, and he was a monk in the early 1800s who studied heredity using pea plants. So what he did was he cut off the reproductive organs of the flower, which would be the part that releases out the pollen, and he instead pollinated these plants by hand, which means he could control which plant mated or pollinated with the other plant. So he could control which characteristics the parent generation had and observe what would happen in the offspring. In his experiment, he started with true breeding or homozygous plants. He took a homozygous purple plant and crossed it with a homozygous white plant. And he found that all of the offspring from that cross were purple. And he scratched his head a little bit trying to figure out what happened to all of these white flowers. So he took all of those offspring in the F1 or the hybrid generation and crossed them with each other again. So a little bit of plant incest going on here. And he found in that F2 generation that he had about 3/4 of the plants with the purple flower and about 1/4 of the plants with the white flowers. He was able to do some statistical analysis on this, and he did this experiment over and over again, and he was able to develop ways that we can predict the offspring. Most of the genetics that we do in this class are called Mendelian Genetics because of the work that Gregor Mendel did. So let's look at an example here with nostril hairs. Pretend that hairy nostrils is a dominant trait. So we will use a capital H to represent the dominant allele and a lowercase h to represent the recessive allele. Notice that we are using the same type of letter for both traits, but the capital version of that letter is associated with the dominant. Now, if the dad is homozygous dominant, his genotype is going to be capital H, capital H. The mom is homozygous recessive, so she's still going to have 2 copies of the gene, one that she inherited from her mom, one she inherited from her dad. But both of those are the same because it's homozygous, but both of them are lowercase because it is recessive for the trait that she has. So her genotype is lowercase h, lowercase h. Now let's think here. If I had a genotype of capital H, lowercase h, so if we had a hybrid, would they have hairy nostrils or not hairy nostrils? Well, they would actually have hairy nostrils, and that's because the dominant form is present. And regardless of how many copies of the recessive are there, if you have that one dominant, it's going to take over. So we will have that dominant phenotype even though there's one copy of the recessive allele present. Now we can predict what the offspring will look like by doing this thing called a monohybrid cross. Now a monohybrid cross is a cross between 2 organisms, but we're only focusing on one trait. We use 4 steps to figure this out. The first step is writing out the equation of the genotypes for each of the parents. Then we put those genotypes around the outside of this Punnett square and solve the Punnett square. Finally, inside each of the boxes of the Punnett square, that will give us the genotypes for the possible offspring. And based off of the genotypes, we can determine the phenotypes or physical appearance of the offspring. Let's look at our baby hair example again. So if we had a dad that is homozygous dominant and a mom that is homozygous recessive, what would their kids have in terms of nostril hair? Well, we start again with the equation, writing the dad genotype times the mom genotype. Then we put those genotypes around the outside of the Punnett square. So each letter is above its own column or next to its own row. I always put the dad genotypes across the top of the Punnett square and the mom along the left side. Honestly, it doesn't make much of a difference. It just makes it look pretty for me. Now the dad puts a capital H over the left column, capital H over the right column. The mom puts a lowercase h by the top row and a lowercase h by the bottom row. Now we fill in our square, just bringing that capital H down, bringing that lowercase h across. So we have the capital H brought down from the dad plus the lowercase h from the mom, and those get brought in to make capital H, lowercase h, which is one possible baby. Now the next box, again, we bring the capital H down from the dad, lowercase h over from the mom, and we get another heterozygous individual. So we'll fill in the other two boxes using that same method. Then the next step is reporting our possible offspring. When we look at our possible offspring, each box is its own possible baby and has the same statistical likelihood of occurring. So inside those 4 boxes, 4 out of 4 are capital H, lowercase h. So 100% are heterozygous. When we look at the phenotype, we would turn that genotype into a phenotype, and we would have 100% of those possible babies having hairy nostrils. Now let's look at another example here with guinea pigs. If a rough coat is dominant over a smooth coat, and we are crossing a homozygous rough-coated guinea pig with a smooth-coated guinea pig, we can figure out the possible genotypes and phenotypes of their offspring using the same monohybrid cross again. So the equation for Pete, the homozygous rough guinea pig, would be capital R, and for Chris, the smooth-coated guinea pig would be lowercase r. Again, we start with our equation with the father times the mother, putting in our genotypes. Then we set up our Punnett square. So we put the father alleles across the top, so he could contribute a capital R, or he could contribute another capital R. The mother could contribute a lowercase r, or the other possible letter she could contribute would be a lowercase r. Now we bring the letters down and move them across to fill in each of the boxes. And then we report out the possible genotypes and phenotypes. 4 out of the 4 boxes, or 100%, could be capital R, lowercase r, which is heterozygous. The phenotype would be a rough coat, because again, one copy of the dominant allele is all it takes to show the dominant phenotype. Now, in summary, you should be able to define each of the following words: genetics versus heredity, gene versus allele, genotype versus phenotype, heterozygous versus homozygous, and dominant versus recessive. You should also be able to explain the law of segregation and the law of independent assortment, and you should be able to solve a monohybrid cross.